Hi friends, today we are going to study about DNA sequencing and that one of the methodology used for the DNA sequencing. So first we will introduce what is meant by DNA sequencing. The determination of nucleotide or ATGC based sequence of DNA, we call it as a DNA sequencing. Okay, so here we are determining the ATGC sequences of DNA. We have since past we have several different methods for the DNA sequencing, like first one Maxam Gilbert sequencing, which is also known as chemical sequencing, Sanger sequencing, also known as dideoxy chain termination method, automated DNA sequencing, pyro sequencing, Roche 454, solid sequencing, and illuminescence. Out of this, today we are going to introduce Maxam and Gilbert sequencing method. So coming to the first method, that is called Maxam Gilbert sequencing or chemical sequencing method. This method was discovered by Alan Maxam and Walter Gilbert. It is generally based on the end labeling of DNA followed by chemical modifications and subsequent cleavage at specific modified base. This base-specific chemical cleavage involves modification of the concerned base, then removal of the modified base from the DNA strand, and finally the induction of strand break at the position of DNA where base has been removed due to chemical modification and subsequent cleavage. So for the base-specific chemical cleavage, we are using different reagent chemicals like dimethyl sulfate, piperidine formate, hydrogen with NaCl, which are able to modify and remove various nucleotides. Like dimethyl sulfate causes the modification and the removal of G nucleotide. Piperidine formate causes modification either A or G nucleotide in the DNA. Hydrogen is able to cause modification at C or T nucleotide, and hydrogen with NaCl is able to modify and remove C nucleotide in the DNA. So like this various base-specific chemical cleavage we are performing with the use of different chemical reagents specific to different bases in the DNA. How these chemicals are modified has been given here in this table. For example, dimethyl sulfate causing the methylation of N7 residue of the guanine, and thereby it making C8 and C9 carbon bond to breakage or to cleavage, and thereby removal of G nucleotide from the DNA strand. So like this, several chemicals they have a individual reaction to remove specific nucleotide in the DNA template. So coming, we will introduce about how method is carried out to determine the DNA sequence. Okay, so first and the most important step in the Maxam Gilbert sequencing is to get a pure quality DNA template for the sequencing. So first step in the DNA sequencing here is to get pure quality DNA preparation. Okay, here one of the DNA template is shown here with double-stranded structure. First step in the Maxam Gilbert sequencing is to label at one of their end, either at 5' end or either at 3' end of the selected DNA template. The DNA template can be labeled at their 5' end by using polynucleotide kinase or at their 3' end by using terminal deoxynucleotide transferase. This labeling is done with the use of radio label P32 molecule, okay, which is used to label at 5' end or at 3' end. Once we have labeled any one of the end, 5' or 3', the two strands of DNA they are separated by gel electrophoresis, and finally the single-stranded one-end label DNA template is subjected to four separate chemical reactions. In which we have added different chemicals which are specific to cleave at a different DNA bases. The first reaction which are able to cleave at A or G nucleotide, in which the piperidine is added. The second reaction contains cleavage chemical specific to cleavage at C nucleotide. Third contains chemical to cleavage at G nucleotide. Fourth one contains which are able to cleave both at C and T nucleotide. Once we have subjected this base specific chemical cleavage, we will get a different size band fragment according to the modification in cleavage site within the DNA template. Okay. After the base specific chemical cleavage, we are subjecting the reaction mixture from all these 4 reactions into different lanes of the same gel, into different veins of the same gel, which are adjacent to each other. Okay, and then we are performing separation with the help of electrophoresis, and then determining the base sequence using autoradiography. The autoradiography is used here to detect the DNA band. Okay, and the sequence of DNA base pair sequence of DNA is determined by reading the band size from bottom to the top of the gel with correlating corresponding reaction specific cleavage. Okay, for example, here the lowest band we obtain in the C well, the reaction well in which the base specific chemical reaction specific to C products has been loaded. So first band we have got in C well, along with similar size band we have got in reaction that is specific to cleavage at T or C site. So if we compare both these sides, the band which is present in T plus C nucleotide but absent in T and C nucleotide, but here the band is present in C nucleotide and also in T plus C. So this band is corresponding to presence of C nucleotide. So first nucleotide determined here is based on the comparing the C well and T plus C reaction. So here the first sequence is C nucleotide. Similarly, we will move to the next higher size fragment in the DNA from bottom. We will get the second fragment, smaller size fragment in the G well and A plus G well. So here the band which is present in A plus G but also present in G. So the second nucleotide is our G nucleotide. Okay. Then subsequently we will determine the next banding size and their position in which way. Okay. So here the next band, our next band is present in the reaction mixture containing specific to T plus C cleavage, and there is no corresponding band present in the C reaction tube. So this next sequence will be our T nucleotide in the DNA template. So like this, from the bottom to the top of the gel, we will read sequentially the band and will determine the reading banding sequence, the sequence of the DNA template from reading the band from bottom to the top of the gel. I hope you have understood what is meant by Maxam and Gilbert sequencing. So once we will summarize once again, that is basic steps: obtaining pure quality genomic DNA, single end labeling of the template DNA with polynucleotide kinase or terminal deoxynucleotide transferase, separation of two complementary strands by electrophoresis, subjecting separated single stranded one end label template into 4 specific chemical cleavage, loading the reaction mixture from the 4 specific chemical cleavage into adjacent lanes of the same gel or adjacent wells of the same gel, then detection of the band by the autoradiography, and finally the detection of the DNA sequence by reading from bottom to the top of the gel, sequential reading of the different bands with their corresponding reaction.